Gas transport. In vertebrates, cardiovascular system serves as the transport system for the delivery of food to tissues and the removal of CO2 from tissues. In lung-breathing vertebrates, blood is the medium of transport of respiratory gases between lungs and tissues. It transports O2 from lungs to tissues and CO2 from tissues to lungs. Only very little of O2 and CO2 gets dissolved in blood plasma. So most of them will be transported in chemical combinations in plasma and RBCs. First, let's learn about transport of oxygen. Blood transports O2 in two ways, in chemical combination with hemoglobin, in physical solution in the water of plasma and corpuscles. In human beings, about 97% of O2 is transported in combination with hemoglobin and about 3% in a dissolved state. 97% of O2 is transported in combination with hemoglobin and about 3% in a dissolved state. So transport of O2 in physical solution. Transport of oxygen in physical solution depends on the solubility coefficient, pressure gradient and temperature. Normally 1 ml of blood absorbs nearly 0.024 ml of oxygen at a temperature of 37 degrees Celsius and a pressure of 760 mmHg. The partial pressure of oxygen inside the lungs is 100 mmHg. So at this pressure of oxygen, 1 ml of blood can carry 100 into 0.024 by 760 is equal to 0.003 ml of oxygen in physical solution. Then transport of O2 by hemoglobin. Hb has all the accomplishments of an oxygen transporting respiratory pigment because it can transport large quantities of O2. It has high solubility, it can take up and release oxygen at appropriate partial pressures, and it can serve as a powerful pH buffer. During oxygen transport, Hb is only oxygenated and never oxidized. During oxygenation, its ion atom stays in the ferrous state itself. It is oxidized to ferric state, its oxygen carrying capacity would be lost. So that is protected with a covering of hydrophobic groups of globin. Then hemoglobin has great affinity for O2 at high PO2 and low PCO2. At a high PO2 in the capillaries of respiratory surfaces or lungs, it will readily accept oxygen and its Fe2 plus ions will loosely and reversibly combine with O2 to form the unstable oxyhemoglobin Hb4O2. This is called oxygenation or oxygenation of hemoglobin. High blood pH or low acidity also enhances the oxygenation of hemoglobin. In this reaction, the ion of hemoglobin stays in the ferrous state, and so the reaction is oxygenation and not oxidation. Don't confuse with these terms. The reaction has been customarily represented as Hb plus O2, HbO2. Since hemoglobin contains 4 heme groups and 4 polypeptide chains, it is more appropriately represented as HbO4 instead of Hb. So the oxygenation reaction of hemoglobin is more appropriately represented as HbO4 plus O2, Hb4O2. The extent to which oxygen gets bound to Hb varies with the PO2. The PO2 at which hemoglobin gets oxygenated is known as tension of loading. This is a new portion and term. The extent to which oxygen gets bound to Hb varies with the PO2. The PO2 at which hemoglobin gets oxygenated is known as tension of loading. Tension of loading. Okay. Each ion atom of it can bind with one molecule of oxygen. Since a hemoglobin molecule contains 4 ion atoms, it can bind with as many as 4 molecules of O2 at a time. The condition in which all the binding sites of hemoglobin molecule are simultaneously copied by oxygen is called 100% saturation of hemoglobin. 100% saturation of hemoglobin. It occurs at high tension of loading at high PO2. Hb4 plus 4O2 gives Hb4O8. Hb4O8. Hb4 plus 4O2 gives Hb4O8. Okay. So hemoglobin saturation and PO2 Perhaps the most important factor that determines how much oxygen can combine with Hb is PO2. Hemoglobin exists in three conditions of oxygenation or saturation, namely unsaturated or unoxygenated, partially saturated or partially oxygenated, and fully saturated or fully oxygenated. In the unsaturated state, it is free of oxygen. In partial saturation, 1, 2, or 3 of its ion atoms may be oxygenated. In the fully saturated state, all the 4 ion atoms are oxygenated. The degree of the saturation of hemoglobin with O2 is directly proportional to the level of PO2. The percentage of saturated Hb in the total hemoglobin is called percent saturation of hemoglobin. Percentage saturation of hemoglobin. And saturation of hemoglobin is a stepwise process. During each step, one of its ion atoms gets oxygenated. Not oxidized, just oxygenated. So the full saturation of hemoglobin takes place in 4 steps as per the following scheme. First, Hb4 plus O2, Hb4O2, Hb4O2 plus O2, Hb4O4, Hb4O4 plus O2, Hb4O6, Hb4O6 plus O2, Hb4O8. So HbO4 plus O2 gives Hb4O2, Hb4O2 plus O2, Hb4O4. Along with that, Hb4O4 plus O2 will be giving Hb4O6. Along with that, Hb4O6 plus O2 will be giving Hb4O8. It has been estimated that 1 gram of hemoglobin can combine with as much as 1.34 ml of oxygen, greater than the content of hemoglobin. Okay, this is the oxygen carrying capacity of hemoglobin. 1 gram hemoglobin contain 1.34 ml of oxygen. Okay, oxygenation of one heme group not only increase the affinity for the remaining heme groups for O2, but also facilitates their oxygenation. This is called cooperativity. It may be probably due to conformational changes in the globin part and is called homotropic interaction. The binding of allosteric modulators at any site of the heme molecule other than the oxygen binding site may lower the affinity for O2. This may be called heterotrophic interaction. So, oxygenation of one heme group not only increase the affinity of the remaining heme groups for O2 but also facilitate their oxygenation. This is called cooperativity. It may be probably due to conformational changes in the globin part and is called homotropic interaction. Cooperativity. It may be probably due to conformational changes in the globin part and it is called hemotropic interaction. The binding of allosteric modulators at any site of the heme molecule rather than the oxygen binding site may lower the affinity for O2. This is called heterotropic interaction. Heterotropic interaction. Okay. Combination of the first heme molecule with O2 increase the affinity of the second heme molecule. In the same manner, oxygenation of the second heme molecule increases the affinity of the third one, and oxygenation of the third molecule increases the affinity of the fourth molecule. Thus, the affinity of the fourth heme molecule for oxygen would be many times greater than that of the first molecule. So, saturation of hemoglobin is therefore an example of a positive feedback. Positive feedback. Hb plus O2 affinity 1 time, HbO2 plus O2 affinity 2 times, HbO4 plus O2 affinity 4 times, HbO6 plus O2 affinity 8 times, HbO8 will be formed. When hemoglobin takes up O2, its 2 beta chains move closer to each other. When it gives up O2, the beta chains move away from each other. It is this shift that determines the shift in the affinity of hemoglobin for O2. In the tissues, PO2 is very low and CO2 is very high. Hence, oxyhemoglobin dissociates and sets free O2, forming deoxyhemoglobin. This is known as deoxygenation or desaturation of hemoglobin. The PO2 at which hemoglobin gets deoxygenated is known as tension of unloading. Tension of unloading. then HbO4 plus O2, high PO2 plus low PO2 at respiratory surface, low PO2 plus high PO2 at low tissue site. HbO4 Hb4 plus O2 gives Hb4O2, oxygenation of Hb, reverse deoxygenation of Hb. The dissociation of oxyhemoglobin is influenced by several other factors also, such as acidity, low pH, and presence of 2,3-diphosphoglycerate or DPG, high body temperature, high PCO2, etc. Under acidic conditions, the equilibrium between deoxyhemoglobin and oxyhemoglobin gets shifted in favor of the deoxygenation process, and oxygen dissociates more readily from hemoglobin. This is called Bohr effect, BOHR. Bohr effect. Okay. Bohr effect. DPG is an intermediate product of glycolysis. It reversibly combines with oxyhemoglobin and alters its structure to release oxygen. So DPG enhances the delivery of oxygen to tissues. The increase of temperature up to a limit enhances the dissociation of oxyhemoglobin. This temperature is maintained by the heat produced during metabolic reactions. Next is about Bohr effect, BOHR. Bohr effect is a phenomenon whereby the acidity or the high level of dissolved CO2 in blood inversely influences the oxygen carrying capacity of hemoglobin and other respiratory pigments. So in an acidic condition, oxygen dissociates more readily from hemoglobin. Oxygen dissociation curve shifts to the right. Most respiratory pigments exhibit a negative Bohr effect. This means that pigment molecules have lower affinity for O2 when the levels of dissolved CO2 in the blood are high, and also that they have maximum affinity when the CO2 level is low. This is an adaptation in which facilitates maximum gas exchange. Bohr effect is significant in three respects. Once again, I would be explaining. This means that pigment molecules have lower affinity for O2 and the levels of dissolved CO2 in the blood are high, and also that they have maximum affinity when the CO2 level is low. This is an adaptation which facilitates maximum gas exchange. The Bohr effect is significant in three respects. It enables maximum intake of O2 by the respiratory pigment in the respiratory surface, gills, lungs, etc. Where the CO2 level is low, enables the maximum flow of O2 from respiratory surface. Once again, I will be explaining this. Bohr effect is significant in three respects, which are it enables maximum intake of O2 by the respiratory pigment in the respiratory surface, gills, lungs, etc., where the CO2 level is low, enables the maximum flow of O2 from respiratory surface to tissues, and enables the maximum delivery of O2 to tissues where the CO2 level is high due to metabolic production, and the oxygen binding powers of pigment is very low. It enables maximum intake of O2 by the respiratory pigment in the respiratory surface where the CO2 level is low. It enables maximum flow of O2 from respiratory surface to tissues, and it enables maximum delivery of O2 to tissues where the CO2 level is high due to metabolic production. That CO2 level is high in case of tissues because of metabolic production of CO2, and the oxygen binding powers of the pigment is very low. Bohr effect is believed to be due to the alteration of the structure of hemoglobin when hydrogen ions bind to it. This structural change may lower its oxygen bonding powers. Acidic condition, low pH, usually results from the presence of lactic acid and carbonic acid in blood. Lactic acid is produced during muscular contraction, and carbonic acid is produced by the dissolution of CO2 in H2O. Later, we'll be dealing about oxygen hemoglobin dissociation curve. The uptake of small amount of O2 by hemoglobin favors the relaxed state of its peptide chains. This in turn promotes the uptake of additional O2. This can be graphically represented by a sigmoid or S-shaped curve known as oxygen hemoglobin dissociation curve, S-shaped sigmoid curve. Oxygen hemoglobin dissociation curve, also called oxygen dissociation curve or oxygen equilibrium curve, is a graphic representation of the affinity of Hb for O2 at different levels of PO2. In other words, it is a graph which represents the relation between the percentage saturation of oxygen carrying capacity of Hb and the different levels of PO2 in the blood. It reveals the actual relation between the PO2 and the formation and dissociation of oxyhemoglobin. Combination of the first heme molecule with O2 increase the affinity of the second heme molecule. In the same manner, oxygenation of second heme molecule increases the affinity of the third one, and oxygenation of the third molecule increases the affinity of the fourth molecule. Thus, the affinity of the fourth heme molecule for oxygen would be many times greater than that of the first molecule. When oxyhemoglobin takes up O2, its two beta chains move close to each other, and when it gives rise to O2, the beta chains move further apart. It is this shift that determines the shift in the affinity of hemoglobin for O2.